Welcome to This Medical Life podcast. These are the stories of medicine with Steve Davis and Dr. Travis Brown.
This is a story of a new test called Metapanel.
Dr. Travis Brown, to the layperson, Metapanel sounds like a multivitamin pill. I'm sure it's not. I'm sure you're going to lead us into wisdom.
We will. This is a new test. This actually leads on to a previous episode we did on fecal microbiome. So there was, I think it was episode 40. Yeah, that's right. Episode, I think it was 41, where we sort of looked at the whole changing of microbiology and actually starting to get, now people will be familiar with this with PCR tests, that we're starting to get more and more targets and we're starting to be able to provide more information. But I want to start this episode actually going back to the 1800s actually.
Of course.
And this takes us to a problem that came out of Italy with bleeding statues.
Oh, okay.
And they were having a problem. Now, this is Italy, this is the Vatican area, this is the Pope, but it's sort of whether this was a divine problem or not. And it's hard to tell whether it was a curse or seen as a blessing that these statues have this redness, this bleeding that was happening. And it wasn't just statues, it was actually communion wafers as well. They started getting red blood spots on them.
Okay, now that is beyond the pale.
And there was quite a lot of discussion about this, but there's probably a skeptic out of there. His name was Bartholomew Bizio, who was a pharmacist, and he decided that there must be some reason that this is occurring. This new phenomenon is going on. Now, clearly he didn't think it was divine, but he went down and he said, I'm going to start doing an experiment. And what he got was some polenta, and he put it under a glass globe to see. And then he did a second experiment where he had suspended polenta in the air.
Oh, how do you do that?
He would have put it on a plate just up in the top. And so he left that for a few days. And what he found when he came back was that these red spots had appeared on the polenta. And his suspicions were confirmed that somehow this was either something being transmitted or something was growing on it. Now, he ended up saying that this must have been some sort of fungus. And somehow this fungus was growing on the statues, on the polenta. Now, it turned out it was actually a bacteria, but he named this fungus, so to speak, or this bacteria, Serratia, after his favourite physicist, who was Serafino Serratia. And the second name he gave it was marcians. Now, that is because of a pigment that it produces that rapidly decays. It's a red. So this is Serratia marcians. Now, today, if we look at that, Serratia is actually the red pigment that occurs in bathrooms. And so it's that really hard bacteria or thing to clean in bathrooms. And that's why you get that redness sometimes occurring, because that's actually the organism. And so Bizio's question is one that faced every early microbiologist and even modern pathologists and scientists and microbiologists. They ask, how do I grow this organism? So he used polenta to try and say, now, if he hadn't used something else, it may not have grown on that. But he was actually confirmed his suspicions that this was something that was occurring. It wasn't a divine intervention, probably much to some people's disappointment at that point in time.
It does put a new light. As primary school kids, I think it was early high school kids, leaving Petri dishes in the boys' toilets and the girls' toilets to see what bacteria was captured.
That's right. We did that experiment when I was in high school, but we did it because they weren't quite sure how to leave it. So we got Band-Aids, which were sterilised, and so you'd put it on the tape and then let the tape grow.
Oh, clever.
Yeah.
What discovery have both of us missed? yes.
That's right. And so they were looking at how do we grow these organisms. Now, we think about it so that simply today, but this was their challenge. So they thought, how do we grow it? And so they used things like coagulated egg albumin to try and grow something. They used starch paste, and they even used aseptically cut slices of potato. And so they were trying all these things. The one that was the most successful, but it wasn't very used, was gelatine. And so you put something in gelatine and grow it up. But what they wanted was a solid media to be able to do it, because then you can actually separate them out. And this takes us to Robert Koch, who in 1860s, he's pretty much almost like the father of one of the fathers at least, of microbiology. And he saw that there was a whole bunch of cows dying of anthrax. And he found that there were microscopic rods in the cattle's blood. And what he thought was, well, maybe that's the organism that's actually killing the cows. And so he took that blood and he injected it into sheep, and sheep got the same disease. And so this led us to Koch's postulate, which was 4 criteria. And it says, if you're looking at an organism, it must be present in disease, but not in healthy people. The disease must be able to be cultured. You can inoculate healthy and cause the disease, and then you can rematch that with the original. And so that was sort of saying this is a way to see, does the bacteria cause the disease you're thinking it's caused? So it has to be there and present in disease, but not in healthy. And if you give it to someone who is healthy, then they get that disease.
A question, though. There are some, surely I've learned through this podcast, there are some things present in both health and disease, but it's the quantity and whether it's active or not.
Well, that's right. But you see, what he was saying is, how do we actually separate it out? So if it's in both disease and in healthy, then it's not going to be causing the disease.
Right, okay.
And so that's where he started to go, well, how do we then grow this up? And so they were using gelatin, but gelatin has a problem, and it liquefies at 25 degrees Celsius. And the problem with that is you want to incubate at about 40 degrees or whatever degrees you're wanting to do to grow this up. And the problem is bacteria has gelatinase, which actually liquefies gelatin. And so if you've got then this soup, it's not really of any use because you can't isolate the organism.
Right.
And so we got Robert Koch's assistant was Walter Hesse, and his wife was Fanny Hesse. And there's a few versions of this story that goes about, but the one I liked probably the most, and I reckon will be the most, well, let's say it's the most accurate, even though we can't say that.
This is not Occam's razor. This is Travis's
This is
test.
my version.
Travis's test.
And Walter was having lunch one day, and in the lunch was a jelly, but the jelly was solid despite it being hot. And he looked at it and he had to ask his wife, what is in the jelly? And it was a Dutch recipe that used seaweed agar, seaweed extract agar. They looked at it and thought this might be suitable for a solid media. And it's become pretty much the basis of modern medicine that you go into any, as
That's
you just
what
said.
they did in primary school or high school.
Any agar plate, it's there because we want a solid media to be able to grow up bacteria and separate and isolate all those bacteria so that we can identify them. So every time you go into anywhere that has an agar plate, that is the foundation of microbiology, and it was because of jelly. And so we've then looked at the last, let's say, sort of 150 years of being able to isolate pathogens. And with this isolation of pathogens, we can actually say this is the organism, using Koch's postulate, that causes disease in people. Now, we've gotten really good at that. We imagine thinking sort of something that's making you either vomit or have diarrhoea. We can actually say, identify the pathogen that's causing that. We are even able now, don't even have to grow it. We can actually identify a virus if it is. And so we're now moving and using that identification, but we've now gotten to a point of transition going from a closed question, where we say, is it this pathogen? To now we're starting to get to the point where we don't need media so much. So now we're talking about having an open question as to how many targets we can actually identify. So people are talking about floras, so microbial floras and stuff. This will be touching on that in our discussion with our coming up guest. But it's actually looking at it and saying, okay, we're now getting so much information. We're doing gene testing, so identifying organisms through their genes. And we're now having to start to move to, I would probably say, partially open questions. What does this tell us about your health or your disease? And Metapanel leads into that emerging question.
Great. Now, Travis, can you pass that open Petri dish with agar in it just over here, please? Dr. Michael Wehrhahn is the Director of Molecular Biology and Infectious Serology at the Douglas Hanly Meyer Pathology. Michael has a Master in Public Health, is an infectious disease specialist, as well as a microbiologist and adjunct senior lecturer at the University of Notre Dame. Dr. Wehrhahn is actively involved in research on clinical and laboratory aspects of a range of microorganisms and is a reviewer for a number of journals. Michael's been involved in researching and validating the new Metapanel, available through Sonic in conjunction with Microba. Dr. Michael Wehrhahn's our guest on this Medical Life podcast. Michael, welcome.
Many thanks. Really appreciate it and look forward to this podcast.
All right. Now, let's just start right at the beginning because I asked Dr. Travis Brown, what is Metapanel? And he's deferred the answer to you. What is it?
Okay. So I guess very simply put, the Metapanel is a fecal metagenomic gastrointestinal pathogen assay. It's a diagnostic assay. It's not a descriptive microbiome test. This is a test designed to identify pathogens in a sample of faeces or stool. And it's been developed over several years to be very, very sensitive and to be very specific, so we're not reporting, incorrect results. So,
yeah.
What techniques does it use to get the results? What are we talking about? We're talking about growth. We're talking about PCR. We're talking about what are we using?
Yeah, very good question. So this is really leading technology, referred to as next-gen or massively parallel sequencing. And it essentially is a genomics test where we are looking at all the DNA in a sample. So if we start to get down into the details of the test, the metagenomics is actually called shotgun metagenomics. And just like a shotgun which scatters shot out and just targets everything in its path, as opposed to a very directed, single bullet, we are doing the same with this particular test. So it's essentially detecting microbial DNA sequences that's in that sample. So everything there that's DNA, we're extracting out of that sample and then sequencing it and identifying what's there. So I guess that's one way to think about it. You could differentiate it from, say, 16S metagenomics. 16S is a conserved bacterial gene, and a lot of the previous microbiome work has really focused on bacteria. But the beauty about shotgun metagenomics is you can actually detect not only bacteria but eukaryotes as well. So your parasites, helminths, protozoa, even DNA viruses and fungi are included in this test. So it really has a very broad scope, and we hope that it will pick up pathogens that may have been missed on first-line PCR and culture tests.
So, Michael, it's the other end of the spectrum of those little jelly dishes we put in the girls' and boys' toilets in high school, isn't it?
Yes, absolutely. A long way from there.
How many organisms and pathogens are we talking about?
So at the moment, we market it as 175 targets in the sense that on our report, we are reporting 100 pathogens, so across that range of different types of organism like bacteria, fungi, parasites, for example. And then there's 15 virulence factors across 8 different types of bacteria, and then 60 antimicrobial resistance genes too, because we're looking at DNA, and we can even pick out sort of the DNA of these resistance genes. So that's how we think about it. But if you break down that, I guess everyone's interested in the different 100 pathogens. So I think we've got 53 bacterial species. So that includes 6 helicobacters, for example, a lot of enterohepatic helicobacters that may play a role. Campylobacters, there's at least 5 or 6 of those. 6 vibrios, for example. We have some sexually transmitted infections, so because sometimes they masquerade as inflammatory bowel disease with severe proctocolitis, such as lymphogranuloma venereum, and so we had those organisms like syphilis and gonorrhea and chlamydia in there as well. And then there's some really weird and wonderful ones that only cause disease rarely, but it's important to pick those up because diagnosis can be very much delayed in these. And probably the key one that I'm thinking about is Tropharema whippleyi, the cause of Whipple's disease. And this, in one of our validation samples, this patient was symptomatic for 6 months before he was diagnosed after a very long list of some very expensive tests, including a couple of PET scans. So.
Now, I'm familiar with a lot of those organisms, but some every once in a while I go, oh, geez, I'm not quite sure. And I work in microbiology. Yes. So these are for gastroenterologists and then general practitioners. How do we explain pretty much these kind of pathogens to people who may not have even come across these organisms in this kind of circumstance?
Yeah, so that's where our reporting is really key to the I guess, benefit of this test. So every report is reviewed and supervised by a pathologist like myself, and we very carefully try to interpret all the data that's received. We people might be concerned, well, we're going to pick up, lots of these different organisms that might be just residing and not causing any problems. That's not been our experience in our validation samples, even though we sent, a significant proportion, about 10% had 3 or more different confirmed targets on culture and PCR combined. The majority, I think, when they actually put this to the real world test, is that this is not the case, and maybe 1 or 2 at most is being detected. But some of these less commonly described ones where we don't culture, for example, like Campylobacter concisus, we tend to then move to a deeper insight by assessing how much is there. So that there is this, because of the DNA, it's sequencing all the DNA in that sample. If you've got, for example, a case of Clostridium difficile colitis, there's not going to be a lot of the normal healthy flora there, and there's going to be a predominance or a significant abundance of Clostridium difficile. And so I guess as a converse of that, if you get a very, very weak abundance, for example, of one of these potential pathogens, we can craft comments that explain that this has only just been detected at a very low level and may well be colonizing flora as opposed to causing pathogenesis.
So with the amount of tests that are going on, you mentioned antimicrobial resistance genes. Is that going to be significant, like if we're looking for someone for chronic disease, chronic diarrhoea, or how will they play a factor in this?
Yeah, that's a really excellent question. And we see that the additive value of these AMR genes really as extra information in future treatment of these patients. So particularly as we maybe find a role for this test in those who are heavily immunosuppressed and prone to developing sepsis from their own gut flora that's been altered. So they have a dysbiotic, abnormal microbiome that's carrying these organisms that are usually more pathogenic, and they often also carry these multi-resistant antimicrobial genes. And so being able to detect that in someone who is in that vulnerable category, we hope will be able to inform empiric antibiotic therapy should they deteriorate in the future. And potentially even play a role in infection control if they are carrying certain genes like, or organisms like Candida auris. This is a multi-resistant Candida that we have in this test that may mean that we can sort of curb the spread of that organism in a hospital setting, for example, where we wouldn't have been able to detect that without sort of more, higher suspicion, for example. So,
is this test expected to expand, or is this something that you're happy with 175 and maybe in years we'll have to expand it then, or is it going to be sort of a gradual thing?
Yeah, Travis, this is really the first offering of this test. We have built on the original accredited test of 11 targets, and now have built it to 175, where we think this will really have some clinical utility for those patients that have undiagnosed ongoing chronic abdominal symptoms in particular. And so we do believe that there are organisms that we're still actively trying to incorporate. Gastrointestinal TB is one of those which can rarely cause disease, but it would be nice to have that in there. But due to the quite diverse lineages of TB, we have to be very careful and make sure we can pick up all those and that there isn't some cross-reactivity with other mycobacterial species that might not be as pathogenic, etc. So that's one. A number of the fungi that can infect patients and even get only diagnosed post-mortem in these immunosuppressed patients. So I'm talking about like the zygomycetes and some of these other molds that can cause serious infection in these patients. So we are hoping to incorporate more as we progress this test, but this is the first offering at this point, and we think it's going to be useful for a lot of patients even at this point.
Is it a series of tests? Is it a series of samples, or is it one sample?
It's just a single sample, and so this sample is the DNA is extracted very carefully in a very highly quality controlled lab that's been awarded this very high level of accreditation, this international standard ISO 15189, which means that you can rely on the validity and the reliability of this test, from pre-analytical, analytical, and post-analytical processes. So this test is really very ready for the market, we feel, and it's been developed and tested in a lab that has been very carefully set up and with investment in the technology that reduces the risk of things like contamination or false positives to the greatest degree, really. So it is just the one test, but it's important to recognise that this is a privately billed test, and for that cost of $345, we are actually it takes days to arrive at a result. Just from all these steps, just the different steps along the way. So the first day of really extracting the DNA, that the next day is all to do with preparing that DNA into the best format, so it can then go on to a sequencer and not waste that sort of sequencing run, because the sequencing runs can last up to 48 hours, depending on how many samples there are. And for example, a flow cell on that sequencing machine can cost upwards of $20,000. So there's a lot of costs involved, and then you have to then process that bioinformatically. That takes another day or so, and then the reporting and interpretation of this test. So we have a turnaround time of 10 business days to sort of provide a bit of a buffer in the system. But generally, the actual process is probably more like 5-6 days at the moment, and expected to improve if we have higher demand of these samples. Rather than having a sequence run every second day, it would be then every day, and then that speeds the turnaround time up as well. But it's not a simple 3 or 4 hour PCR test, if you like.
Yeah. Well, it's a labour-intensive pathologist report as well. I've never seen pathologists put their hands up so quickly to say, "I will create this 3 or 4 page report." That's I have seen this here.
Speaking of the report, it's 8 pages with roughly 6 pages of lists of organisms and some information about those. But the front page is where the pathologist really tries to guide treatment and provide an overall interpretation and recommendation that includes references. The last page is interesting too, because not only have we sort of reported the DNA and the genes and virulence factors, but we're also providing an assessment of the microbiome gut health, if you like, and these are accepted measures of microbiome health, like alpha diversity as measured by the Shannon index, and species richness, which we know that more and more studies in conditions like inflammatory bowel disease, irritable bowel syndrome, cardiovascular, metabolic conditions like diabetes, even conditions like depression, they all seem to have this reduction in diversity. And the different organisms might be a bit different, but overall the diversity seems to be reduced. So even if we don't detect a pathogen or AMR genes, we can often provide some information about the health of the microbiome, which then can be taken on board by the patient, because it's not difficult to change your microbiome or improve your diversity by altering your diet, in fact. And I guess the more diverse your microbiome is, it often relies on a more diverse diet. So if you can broaden and increase the number of different foods you're eating each week, particularly from a plant-based origin, then that's going to really stimulate the growth of these healthy bacteria in particular that thrive on these, normally indigestible starches and carbohydrates.
Michael, let's just give that a moment to digest. We'll come back and we'll focus on those clients and patients we might be using this test for.
Sure, great.
Continuing our conversation now with Michael Wehrhahn. Michael, I alluded to thinking about the patients. Who are the ideal patients for this test?
The key patients that we think this test will find use in are those with chronic abdominal symptoms, so bloating, abdominal cramping, pain, plus or minus, loose, frequent bowel motions, so chronic diarrhoea, and they've been through the usual tests like PCR and PCR multiplex tests and culture tests. For example, in our lab, we always do both because that sort of enhances the pickup of these pathogens. And so if there's still a suspicion of infection as the cause of these symptoms, then I think this might be a worthwhile test to consider those patients for. So they're the group that just have undifferentiated abdominal symptoms. I guess a second group are those with already diagnosed inflammatory bowel disease. And these patients often already have this reduced gut microbial diversity. And because of that, they are at greater risk of picking up a pathogen, if you like. There's that less pathogen colonization resistance, if you like. So I think that group will also find a potential role for this test. Sometimes those patients have flares of their IBD, and if they don't find an infection on the standard sort of diagnostic traditional approaches, then this test might actually provide some insight rather than increasing the immunosuppression in these patients, which might worsen potentially that infection. We can actually diagnose or more confidently exclude the possibility of an infection in those patients. So there is another group that we think it may find a role in, and these are the patients who are immunosuppressed. They might not be gastroenterology patients, but they're on immunotherapies and other immunosuppressant therapies that make them more vulnerable to the unusual type of infection, CMV colitis, for example, or these fungal pathogens. And I guess another group too are those that have come from overseas or lived in countries where there are more parasites and helminths or worms, and it's not easy to diagnose worms on this on our traditional testing approaches. These eggs are infrequently released and not in high concentrations, and often get mixed in the stool. But when you're sort of breaking down the DNA and looking for DNA of these helminths, in fact, we sent about 7 or 8 of these stools that didn't have anything seen on them as part of our validation, but we sent them because they had a strong clinical history of worms. And the common worm that we see in our community is pinworm, and these are hard to find from stool. But of all those samples with that strongly suggestive history, we picked up pinworm DNA in those. So, and that was confirmed on a subsequent targeted PCR helminth assay. So we're not sort of presenting false positives. These are confirmed on additional tests. So I guess that they're the sort of 3 or 4 groups that I think clinicians can think about when they're seeing these patients who really are struggling with a potential infective diagnosis. That immunosuppressed group, the AMR genes, may play a role, and just provide them that extra bit of information in terms of subsequent gram-negative sepsis should they become unwell. And similarly, there's evidence to support that those, for example, on immunotherapy or like these checkpoint inhibitors in metastatic melanoma, for example, with reduced gut diversity, they respond less well to that. So having a good science-backed metagenomic assay like this that can give you a fairly confident information about that diversity in that patient, that might also play a role down the track.
You mentioned that you identified pinworm with the shotgun sequencing, but then confirmed that on PCR. Is there a difference in the sensitivities between the 2 techniques?
The PCR is a very, very sensitive technique, almost to the point where some of these organisms like Aeromonas, if they're detected at a very late stage, very high CT value, do they mean much, or are they just colonizing organisms? So certainly the PCR is probably, we would say, that the gold standard in terms of sensitivity, because you can amplify DNA a billionfold. But the beauty about this shotgun sequencing is that if you increase or pick a depth of, so that the number of reads in this assay is 16 million reads, and that can be increased and to increase the depth of coverage. So the more DNA you're actually sequencing, you potentially can get it up to very high levels. And so what we found with our samples that were actually hand-picked in terms of finding those that were really at the threshold of detection by PCR. For example, the Yersinias had CT values in the over 40, and they were all picked up by this assay metagenomically. So we feel that the actual current number of reads is about right, and that the depth is adequate to pick up, at an almost an equivalent level of a PCR, if you like. But some of the organisms in this, for example, didn't quite perform as well as PCR, but was close.
Now, is this a test targeting specialists or general practitioners or both?
At the moment, we feel that probably the greatest utility is by those who are gastroenterologists and have been referred the difficult patient with chronic symptoms. So we have approached that group first off, but we do believe that ultimately the general practitioner group, and they can order this test just as easily. It's simply ordered by just requesting the Metapanel on a standard request form, but there are actually Metapanel requests as well that make it potentially a little bit easier to order for you. But so yes, specialists, GPs, a medical person is the ideal person, because if there's a pathogen that perhaps needs treatment, like a helminth found, then that doctor should ideally be the one to then provide that treatment.
Now we're starting to verge in the area where we're not sure about some of the implications of some of these organisms. Are we expanding the grey zones in this area sort of to be, you'll get a result that's like, we think this is significant or may not be? Are we venturing into that area?
You can look at it in a different way. I think we now are at the point in microbiology where we can use this greater amount of information to actually separate some of these, sometimes referred to as pathobions. These are potential pathogens, perhaps in the right environment. So if we can show that the diversity is low and that there's a particular high level of detection of one of these putative pathogens, like the Campylobacter concisus, for example, which in one big Danish study was shown to be associated with microscopic colitis, or it was associated by about twofold that the likelihood of picking up subsequent microscopic colitis in that group. So I think we're at a point where we have this extra amount of information, and this is a test that could potentially be used in the research setting to look at these different patients, some with a particular clinical syndrome like IBS or IBD, or those patients that just have Campylobacter concisus, and see what's special about those patients and try to diagnose, or more firmly understand that pathogen from a clinical perspective. I think because we are only reporting these pathogens, there's the possibility to drill down into the data and actually look at, well, all those patients, all that subset actually they all might have something in common, and it might be a particular pathogen that just has a code name rather than a full proper sort of name and species. So, this is how we potentially can discover new causes of, common syndromes or even less common syndromes.
Now, are there any limitations or cautions doctors should know about regarding Metapanel?
The main limitations are, as we've already discussed, that potentially we may have to use clinical judgment, and if you don't find an explanation, but we find one of these possible pathogens, then we, have to consider and balance the potential of benefit from a short course of treatment targeting that particular organism, or just waiting and seeing, or exploring other avenues. So I think we've really highlighted that is one of the key issues, but we hope to provide that guidance in the overall interpretation on the front page. The other limitations are that things like virulence genes and AMR genes are much smaller than whole organism genomes, if you like, in the sort of millions of bases. We're talking 1,000, around 1,000 bases for these genes, and so if it's a bit like finding a very small needle compared to a very big needle in a haystack, if you like. So, and so it requires a greater amount. So if you compare the relative abundance of the test on the pathogens, we're looking at being able to pick up, so at a level of 0.0001%, versus 0.01% for the sort of virulence and AMR targets. So you need a bit more starting to confidently exclude those. But it's still, I think, useful information if we do detect that with confidence. That's one aspect, yeah.
There is an area that is starting to emerge, fecal transplant. And is this a test to consider for donors and recipients, or is that something completely separate?
The fecal transplant, I think we've had a lot of interest for people that do these tests, and they're interested in being able to potentially use such a test pre and post transplant. But at this point, we don't really know, and I think it really has to be relegated to this research realm, if you like. If you think about donors, it may well be a good test to actually more comprehensively screen those donors. At the moment, I guess there are the tests to screen those potential donors using our traditional approaches, which may miss some people. So you could argue that such a broader test as this, that includes even multi-resistant organism screening, might be the way of the future. Because certainly some people, there are case reports of people being transplanted these multi-resistant organisms that then can have issues down the track.
Certainly, if I was a recipient, I would be wanting my donor to have this screening. There's not much more intimacy you can have between two humans on this planet. Finally, Michael, any last thoughts or advice that you have about Metapanel for our listeners, particularly GPs, but the specialists listening as well?
I guess I've mentioned that this test is still in its early stages, so we do expect that this will become a more refined and more useful test as the target list potentially increases to more broadly cover all the documented pathogens. Even though we market this as a 175 target, the pathologists actually review in total 360 odd targets. So a lot of those are sitting in this research sort of area where we're not confident of calling those. One example I think many GPs are aware of is blastocystis and diantomoeba fragilis. Blastocystis is, in some reports even thought to be a marker of gut health. So the jury's really out in terms of whether it does cause symptoms. And so in this assay, we have 7 or 8 subtypes of blastocystis. We may be able to say, well, actually in this patient, nothing was found, but in the supplementary report we have access to, there was a high level of this particular blastocystis. And maybe based on the genotype, this can actually provide some information down the track. For example, the diantomoeba fragilis is another one that unfortunately, because there is no whole genome in the literature to sort of build this test around, we don't have that capacity, even if we wanted to include that in this test at this point. But that's another pathogen that we might include in a supplementary if that genome is obtained down the track.
On that note, Dr. Michael Werhan, thank you very much for being part of This Medical Life.
Oh, you're very welcome, and all the very best. Thank you.
This Medical Life is recorded in the Talked About Marketing Studios in Adelaide. For show notes and more information about the podcast, visit thismedicallife.com.au. You can contact the hosts on social media. Dr. Travis Brown can be found on X. His username is @Dr. Travis Brown. That's Dr. Travis Brown. And Steve Davis can be found on LinkedIn. Go to linkedin.com/in/TheRealSteveDavis. This has been a PathNotes Proprietary Limited production.